Let's study 9th standard ICSE Biology Chapter 2, Cell Organelles. Let's study the table which has totally 15 parts of the cell. We'll study their characteristics and functions. Starting with the cell wall, which is only found in plant cells. It is the outermost layer in plant cells. It is non-living. It is rigid and freely permeable. Anything can enter or exit, even large molecules. It is mainly composed of cellulose, which we can't digest even if we eat it. But it's useful as a fiber for preventing constipation. Its function is to give rigidity and shape to the plant cell. It allows substances in the solution to enter and leave the plant cell, please mention that, without any hindrance. Hindrance means hurdle. And it provides protection to the plant cell. Well, a differentiate between cell wall and plasma membrane question can be asked. Plasma membrane is also called cell membrane. It is the outermost in animal cells. It lies next to the cell wall in plant cells. It is very thin, flexible, living membrane. So this was non-living and this is living. Another difference is that it is freely permeable and this is semi-permeable. Semi-permeable means that only small molecules can enter it. By the way, it's also selectively permeable. That means it can change the size of its pores to allow larger molecules to enter when required. It's a living thing after all. Another difference here is that this is made up of lipoproteins. Lipo means fats. Chief functions. Well, it separates the contents of the cell from its surroundings. It regulates the entry of certain solutes and ions in and out of the cell, and it maintains the shape of the cell in animal cells. Next, the cytoplasm. All the parts together inside the plasma membrane, excluding the nucleus, is called cytoplasm. It contains a mixture of water and soluble inorganic and organic compounds and even various organelles. Function, well, first of all, it has so many organelles which have their individual different functions. All metabolic activities, that is all biochemical activities, occur in it. It is a medium for the initial steps of respiration, that is the production of pyruvic acid, which is anaerobic respiration. Anaerobic means without oxygen. So respiration has two major steps. First of all, the glucose is broken down into pyruvic acid. This process is called glycolysis. You will study this in detail in another chapter of respiration. But we need to know that this step is anaerobic, does not require oxygen. It happens in cytoplasm. And the next step is aerobic. It requires oxygen, where the pyruvic acid is broken down into carbon dioxide and water, the waste products, and energy is released in the form of ATP. And can you guess where does this step happen? It happens in the mitochondria. So the anaerobic part happens in the cytoplasm for the production of pyruvic acid. Whereas in the mitochondria, the powerhouse of the cell, we have the aerobic part, which requires oxygen. So its function is release of energy from the pyruvic acid. Where was the pyruvic acid created? In the cytoplasm, from glucose. And that is further broken down, and energy in the form of ATP is released and stored by the mitochondria. Another function of mitochondria is synthesis of respiratory enzymes. So whatever enzymes are required for the reaction are produced by the mitochondria itself. Some characteristics of mitochondria, various shapes, but usually sausage-like, as you can see in the diagram. It is a double-walled organelle, and the inner wall is thrown into folds called cristae. Can you see that we have very many folds inside it? They are all cristae. That increases the surface area, and so reactions can happen more efficiently. Surprisingly, they have their own DNA and their own ribosomes as well. So they are semi-independent. They do obey the instructions of the nucleus, but they can multiply themselves on their own. It seems that billions of years ago, they were independent organisms. They can produce their own energy, they can reproduce on their own. But today they are a part of a team called cell, because united we stand. The next organelle will study is endoplasmic reticulum, which is like the highway of the cell. It's an irregular network of double membrane tubules. It is continuous with the plasma membrane and the nuclear membrane, which means it connects the cell membrane to the nucleus. It may be smooth or rough. In the diagram you can see it is rough because ribosomes are attached to it. Its function is, it is a supportive framework for the cell. Imagine a three-dimensional cell which is like a soft jelly. It needs some supportive framework, which would be the ER. And also, ER helps in the synthesis and transport of proteins and fats. So the ribosomes attached to it will prepare the proteins, but who will transport it to the various other parts of the cell? The ER will. The next organelle we'll study is Golgi apparatus. In plant cells, they are called dictyosomes. They are stacks of flattened membrane sacs, Stacks means piles of them, as you can see, one on top of the other. They consist of tubules, that is cisternae, vesicles, and vacuoles, which are quite similar to each other. So vacuoles are mostly formed from Golgi apparatus. The function of Golgi apparatus, the packaging center of the cell, is synthesis and secretion of enzymes, hormones, etc. And also formation of acrosome of sperm, but only in animals and human beings. So what is this acrosome and what is its use? Well, during sexual reproduction, we know that a sperm will have to fertilize an egg cell. So once the fertilization or penetration happens, the 23 chromosomes of the sperms will combine with the 23 chromosomes of the egg cell in the human being, and a zygote will be formed with 46 chromosomes. But in order to enter the egg cell, we need a chemical to make a hole there, and that chemical is the acrosome. So the fastest sperm, which comes first, will get this opportunity to fertilize the egg cell, and the remaining millions of sperms will just wither away. The next organelle is ribosomes. They are small granules either scattered in the cytoplasm or attached to the ER, making it look rough. Now, they are single-walled, unlike the other organelles like mitochondria, which are double-walled. They are quite dense, spherical bodies, and they are made up of RNA, ribonucleic acid. Its function is protein synthesis. But to synthesize proteins, it needs a recipe. Well, that recipe is mentioned in the DNA. Next organelle, lysosomes. They are membranous sacs budded off from Golgi bodies. So, like vacuoles, even they are born from Golgi bodies. They contain up to 40 different types of enzymes, and they have 4 functions. First, intracellular digestion, which is inside the cell. They destroy all foreign substances. They use these enzymes to digest anything foreign which enters the cell and protects the cell. Third point is a give reason. If the cell is old or injured, then lysosomes will rapidly destroy the organelles, destroy the cell, so that new cells can take its place. We should not let injured and old cells exist. What if they turn cancerous? So by doing this, the lysosome itself gets destroyed. It sacrifices itself to save the organism. It's called a suicide bag of the cell. And another function is that it digests cartilages during formation of bones. The next organelle is centrosome, which is found only in animal cells. It is a region surrounding the centrioles located near the nucleus. So centriole is not an organelle, but the centrioles, along with these microtubules called asters around it, together is called a centrosome, only in animal cells. During cell division, these asters form spindle fibers. So the function of centrosome is to initiate and regulate cell division in animal cell. Spindle fibers look something like this. You can see from the centrioles, the spindle fibers are being formed so that they can pull the chromosomes in the opposite direction, ultimately helping in cell division and preparation of daughter cells. You will study cell division in detail in the 10th standard textbook. Next, plastids, only found in plant cells. There are 3 kinds of plastids. The most common is chloroplast. You can see this is one chloroplast. Inside you see those green colored discs. They are called thylakoids, and in the walls of the thylakoids we have a pigment called chlorophyll. That is why it's green in color. They help in capturing sunlight and do photosynthesis. The plastids are double membraned. They have a proteinaceous matrix, that is the fluid filling them is proteinaceous, made of proteins, and like the mitochondria, even chloroplasts contain their own DNA. So they are also semi-independent. So the 3 types of plastids now. First, chromoplasts. They are colorful. They impart color to flowers and fruits. Xanthophyll pigment is yellow and carotene is red. And this color helps in pollination of the flower and dispersal of the seeds because it attracts insects and animals. Chloroplasts help in photosynthesis, of course. And leucoplasts, they don't have any color. They're colorless. Their function is to store starch. Example, potato has a lot of leucoplasts. Now the pigment called anthocyanin gives blue-violet color to brinjal and other fruits and vegetables. But they are not present in the chromoplast. They are present in the cell sap, which is in the vacuole. Next, the most important organelle, the nucleus. It is the largest cell organelle, considering that vacuoles are not living. Nucleus is mostly spherical and dense. The nuclear membrane has pores, which allow substances to enter and leave. It contains network of thread-like structures called chromatin fibers, which contain DNA. And during cell division, these chromatin fibers condense to form chromosomes. And it is filled with nucleoplasm. Functions? Well, the first and the second point are actually the same, so they should be together as a first point, and it should be a give reason. If you remove a nucleus, the cell dies. Why? Because the nucleus regulates all cell functions. And the second function is that it has chromosomes, the bearers of genes, and the genes control hereditary characteristics. Genes are made up of a chemical called DNA, of course. One gene has a recipe or a code for one protein. In the human cell, we have totally around 18,000 genes. Nucleolus, it's a dark spherical body in the nucleus. There may be two as well, called nucleoli. It produces ribosomes. So, since ribosomes do protein synthesis, you can see nucleolus is directly involved with protein synthesis. It also forms RNA, which is also required to do protein synthesis. They also dictate the ribosomes to synthesize proteins. Chromatin fibers. Well, during the resting stage, they are chromatin fibers, but during cell division, they condense to form chromosomes, and then they look something like this. Here, the chromosomes. Function, well, as I said, they have the genetic information which is passed from parents to offsprings. Now we have two more. They are non-living. We can't even call them cell organelles. Let's just call them cell parts, because organelles have to be living. Vacuoles. They are clear spaces with water or other substances in solution. Plant cells have larger vacuoles, while the animal cells have smaller ones, if at all present. And they are covered by a covering called tonoplast. Functions. Well, they store water and other substances like food and waste products. They are like the go down. They give turgidity to the plant cells, that is some kind of rigidity, by pressing against the cell wall. It's like a balloon is filled with water, so it's quite turgid or rigid. If you remove all the water, it becomes flaccid. Same thing happens with plant cells. And it contains some pigments like anthocyanin, a blue-violet color. And finally, granules. They are small particles, crystals or droplets. They are just food for the cell. Example, starch in plant cells, glycogen in animal cells, and even fat droplets. Hi students, this is AJ sir. If you like this video, press the like button. If you would like to enroll for my online test series or online lectures, email me or message me on Instagram. Check the description for more information.